everyone. My name is Daniel. I'm one of the graduate students of Dr. Bonos. I'm very happy to present our work in this remote way. My presentation is titled Inheritance of Summer Patch Resistance in Hard fescue. Hard fescue is an important species for low maintenance landscape with dry soil and low fertility levels. Summer patch is a root disease, and symptoms of summer patch disease are typically described as distinct sunken and depressed zones of necrotic turf with a irregular shape and are often referred as the frog eye symptoms. Susceptibility to summer patch disease hinders the popularity of hard fescue. So, in this work, we wanted to determine if the summer patch disease resistance is controlled by major genes, and we also want to determine the inheritance of summer patch resistance in hard fescues and provide insights of efficiency and efficacy on the hard fescue selection method. In this work, three summer patch resistant clones R7, R8, and R10, and three summer patch susceptible clones S5, S6, and S11 of hard fescue were identified and selected from a preliminary trial and were crossed in a diallele crossing design in the spring of 2015 for a total of 15 crosses and reciprocals. In the summer of 2017, 100 progenies from each of the 15 crosses and reciprocals were established in a mode-spaced plan trial, Trial 1. An exact replication of the trial was established in the summer of 2019, Trial 2, with all the clones vegetatively replicated. Both trials are in Rutgers University Horticultural Farm 2, located in New Brunswick, New Jersey. All populations, as well as selected parental genotypes, were arranged in a randomized completed block design with 4 replications. 3 cubic centimeters of summer patch inoculum were placed under all plants prior to planting with a mixture of both an M. Meyeri fastuosa isolate and an M. poa isolate for both trials. We took visual rating on a 0 to 10 scale to assess the hard fescue individual plant health in year 2018, 2019, and 2020, where 0 indicated completely dead, 1 indicated nearly 100% summer patch damage, and 10 indicated nearly 0% summer patch damage. In the top left figure, we notice the frequency distribution of the row disease visual rating data shows 2 peaks in the bar chart, which suggests that the summer patch resistance on hard fescue may not be a typical quantitative trait. To determine this, we applied major gene indices proposed by Carlin. MGI is a class of indices for indicating the presence of major genes. We calculated the MGI value significantly larger than 1, which indicated that summer patch resistance in hard fescue is most likely controlled by a few major genes. In the field picture, in general, we can see a clear pattern. The progeny from resistant crosses are less summer patch symptoms than the progenies from susceptible crosses, with the progeny from resistant and susceptible crosses in the middle. We used the visual rating data to form the figure 5 of mean summer patch rating at the top right corner. We observed a gradient here. The Progenies from the crosses of susceptible parents generally showed more summer patch disease. The progenies from the crosses of resistant parents generally showed less summer patch disease, while the progenies from the crosses of resistant and susceptible parents were generally in the middle range. To its estimated the estimated maternal and paternal effects on summer patch disease for each parent were present in the bottom right figure. Parents R10 and R8 are significant positive have significant positive breeding values. Parents S5 and R7 have a significant negative breeding value, while parents S11 and S6 are considered to have neutral breeding values. It's interesting to point out that even though the parent genotype R7 is confirmed to be resistant to the summer patch, it has negative genetic effects to its progeny towards summer patch resistance. The genetic variance and total phenotypic variance were calculated and shown in the bottom left figure below. Compared to the additive genetic variance, the dominant genetic variance is relatively small, and we observed a moderate narrow-sense habitability value 0.67 with a standard deviation of 0.01 and a large broad-sense heritability value 0.96 with a standard deviation of 0.01. The moderate narrow-sense heritability suggests that we should be able to improve summer patch resistance in hard fescue via selection and breeding. However, the variability in breeding values of some of the parents suggests that the pre-selection of parental genotype is important to guarantee the efficacy of the breeding program. Thank you very much for listening.